Impurity limits justification is the topic for the present video, and this video is very important to every professional who is working in the pharmaceutical industry and also in the academics. All pharma professionals should know the science behind the demonstration of safety and efficacy of the pharmaceuticals, and impurity limits is one of them. Generally, impurities are of different types like organic impurities, inorganic impurities, then residual solvents, and the nitrosamine impurities. Out of these, the video is regarding organic impurities. In other words, these organic impurities are known as related substances. These may be the degradation products or these may be the process related impurities. But for pharmaceutical drug product, mainly the degradation products are considered. The impurity limits are set as per the monographs, as per the stability data and ICH guidelines. But if the impurity level crosses this safe limit or limits provided by the ICH guidelines or the regulatory guidelines, then there should be qualification. So, how to justify the higher specification limits is the aim of this video or objective of this video. Let's start with some basic informations. How to justify higher specification limits for the impurities. Many of the professionals do this activity in advance, or sometimes this activity is required to be done after collection of the exhibit batch data, because we don't know whether any impurity will increase or sudden increase will be there or not. Always have stability data in hand before providing the impurity limits. Also perform force degradation data and understand the impurity degradation or degradation impurity formation pathways. Whenever you are going to justify the higher specifications, the activity starts from a risk assessment. Risk assessment should evaluate the higher impurities effect onto the product quality, safety and efficacy. What happens if the impurities are higher in their presence in the product? You can use ICH Q9 principles for risk assessment. ICH Q9 guideline is a quality risk management guideline which can guide you for risk assessment. Then safety data is required. You can go for toxicological studies and you can evaluate the safety of the impurity or the degradation product. Generation of the data for safety and toxicity is called as qualification process. Gather information on the threshold of toxicological concern or permissible daily exposure, that is PDE, as per ICH M7 guideline or ICH Q3B guideline. These guidelines are there for the impurities. Literature evidence can be provided. You can search for published studies or regulatory procedures which show the similar impurities at higher levels in the approved products. That means from literature you can collect the data and you can study whether the literature is showing the similar impurities at higher levels in the approved products. But always understand that this literature should be very authentic and should be uh, able to provide you the correct information. So include peer reviewed studies or references to justify the safety. If you found some information in the specific literature you should evaluate whether you can rely on that study or not. Then for higher specification justification, clinical relevance is the requirement. So ensure that clinical trials have evaluated the formulation containing these higher impurity levels. That means if there is a clinical trial conducted with the formulation containing the impurity at these levels or higher levels than your specification, then you can say that these impurities are qualified with this level, and that will be a strong justification. Provide evidence that no adverse effects or reduced efficacy occurred at these levels. For example, if the impurity A limit is 0.5% for your product and you are observing it at 0.7% or 1%, then you need to justify. And justification needs thorough evaluation of the impurity and qualification. Then next step is the analytical data. Demonstrate that the impurity profile remains consistent under stressed and real-time conditions. Show impurity levels are reproducible and controllable during manufacturing. This is very important point to understand. If your impurity is getting increased at accelerated condition or stress condition, but it is not getting increased at the recommended storage condition or real-time condition, then it means that that impurity is not getting increased and you can go ahead with the real-time data or the recommended storage stability data, because some products are not stable at the accelerated conditions. So you can use RT data and go ahead. Also, if the RT data is showing increase in the impurity level beyond your specification, then you need to justify. That time, show the impurity levels are reproducible and controllable. It is not like that only one batch is showing higher impurity level or only one pack is showing higher impurity level and there is no effort taken by the applicant to control it. Show your efforts that the impurity levels are reproducible and these are controllable during manufacturing, through packaging, through storage recommendation, and also through proper care during the manufacturing. For example, if your product is not stable under the whole time period of 1 month or 2 months, then your manufacturing process should indicate that do not store the in process material beyond certain time range. That is the uh, manufacturing control. Then regulatory communication. Once you identify that the product will be with a higher impurity level beyond the justification range or it is a degradant, then you should provide the detailed justification in your regulatory dossier, and it is in the CTD module 3. Or you can communicate with the regulatory authorities once you understand that the impurity levels are higher. So you can engage with the regulatory bodies proactively for their feedback and opinion on the higher impurities, so they can guide you. Determining toxicity of the impurities. You can do the qualification by some methods as given in the guidelines. Structure activity relationship, it is generally called as SAR activity. This SAR uses the computational models to predict the potential toxicity of the impurities based on the chemical structure. Direct software is there, Topcat is there, and some others are there for the SAR relationship determination. Conduct genotoxicity studies, conduct repeat dose toxicity to understand the toxicity limits. Genotoxicity studies involves Ames test, and nowadays enhanced Ames test is there for genotoxicity determination. or in vitro genotoxicity studies are there for the genotoxicity potential. These studies are for any impurity suspected to be genotoxic. It is related to the nitrosamines. So generally these studies are done for the nitrosamines also. If the structure is unknown, then you should make it known and then do the studies. Repeat dose toxicity study if the impurity exceeds the TTC. Perform animal studies to evaluate toxic effects from repeated exposure. Generalized TTC limit is provided in the ICH M7 guideline. If your impurity is below that TTC range, then give the TTC limit, and if it is not, then go for the animal studies. Published safety limits. Refer ICH M7 which outlines acceptable levels for the impurities with known toxicological data. ICH M7 guideline provides you the levels which can be accepted. You should have the toxicological data for the impurities. Databases are there like ToxNet for the safety references. This is the information for toxicity assessment. Now determining the toxicity of impurities by PD calculation. PD is permitted daily exposure. Determination of PD involves hazard identification by reviewing all relevant data. Then you need to identify the critical effects. Then determination of the no observed adverse effect level is done. That is NOAEL, no observed adverse effect level, of the findings that considered to be critical effects and use of several adjustment factors to account for the various uncertainties. And this PD will give you the calculated limit. Use PD limits to establish the maximum acceptable daily exposure using the formula as given here. PD is equal to NOAEL into BW divided by F into UF. BW is body weight. It is considered as 50 kg as a generalized body weight for adults. F is bioavailability factor and UF is uncertainty factor to calculate the PD. Then practical steps for justification. Practically how the steps are there. Collect the data, gather impurity profile data through force degradation studies. You will know that what are the possible impurities and what will be their levels and how these impurities are formed. What is the sensitivity of the molecule? For example, force degradation studies will give you the information for acid degradation, base degradation, peroxide degradation, water degradation, heat degradation. So you can identify by which mechanism or by which factor the impurity is formed. Then run the toxicological assessment. Use computational tools and lab studies for toxicology determination. Consult the experts. Various experts are there in the field who can guide you for the impurity level justification. So work with the toxicologists and regulatory consultants to refine the justification. Tools and guidelines. So various guidelines are there. Guidelines are available from ICH, from USFDA, EMA, ANVISA, and many other regulatory bodies. ICH Q3B provides the guideline for impurities in drug products. ICH M7 is for assessment and control of DNA reactive mutagenic impurities. TTC tools are there for predicting the acceptable impurity levels, and softwares are there for toxicity predictions. Softwares like Derek, Topcat, ACD Tox suit for the toxicity prediction. You can use these uh, softwares to predict the toxicity limits or higher impurity limits. If you find that the impurity limit is safe, then you can justify it and you can give the new specification limits. If you qualify the impurity at 1% or 1.5%, you can give the qualification limit for the impurity which is you found above the specification limits. For example, if you are getting some degradation product in your formulation, and if you have, for example, 0.5% limit for the impurity as per the specifications or as per the guidelines from ICH or monograph, but you have done the toxicological studies and you qualified it at higher limit of, for example, 1% or 1.5%, then directly you can give that limit, and you should try to control the impurity below that level. Your efforts should be there to control the limits or to control the impurities under the limits. I hope this video will help you to understand justification for the higher impurity specification. If you have any queries, you can mail me. I have given my mail ID, or you can comment also. Now you can watch my videos in your native language or the language you understand better by changing the subtitle language. Many languages are available, and YouTube has provided this auto dubbing option for my channel. Thank you for watching the video.